Hello, everyone, and welcome to today's webinar, Applications of Surface Plasmon Resonance, SPR, in HIV-1 Nef inhibitor discovery and PROTAC development. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Reichert Amtech. To learn more, click on the Amtech logo below. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Thomas Smithgall, Professor of Biochemistry in the Department of Microbiology and Molecular Genetics at the University of Pittsburgh School of Medicine. Dr. Smithgall, you may now begin your presentation.
Okay, thank you very much. And thanks for this opportunity to tell you a little bit about our drug discovery efforts targeting the Nef protein of HIV-1 with some emphasis on our use of SPR, which was core technology in this work. Okay, just on background, I'll give you a little update on the status of HIV globally. So it remains as a global pandemic. Currently, close to 40 million people still live with HIV, and 1.3 million new infections continue to accumulate every year. I'm sure many of you are aware that there are potent antiretroviral drugs and combinations that suppress HIV. However, none of these drugs are able to eradicate the virus, and chronic therapy, along with chronic infection, can promote drug resistance. So there's still a big effort to find new drugs that can clear the virus. So the focus of our work is a small viral protein known as the Nef protein. Nef is encoded as one of the HIV accessory factors. It's important for enhancing infectivity of the virus, promoting its life cycle and spread in infected individuals. Importantly, it allows infected cells to hide or escape detection by the antiviral immune response, and it does this in part by downregulating MHC-I, which is essential for HIV antigen display. And other work has shown that this molecule, this viral protein, is essential for progression from a state of HIV infection to full-blown AIDS. Structurally, sorry, I forgot this was here. So Nef inhibitors have the potential not only to suppress the virus, but also to restore the host immune response as part of a strategy to clear the virus. Okay, so a little bit about the structure of Nef. Although I don't, sorry, I don't have a pointer, but if you imagine the cell membrane at the top of the screen, Nef is a myristylated protein, and that promotes its association with the membrane on the inside of the cell. It has about a 60 amino acid anchor domain, a folded globular core, and then an internal loop. And each of these features are responsible for interactions with host cell proteins. So the way we first got interested in Nef is due to its ability to bind and activate host cell kinase molecules. These include members of the SARC protein kinase family, and in particular, a member of that family expressed in macrophages called HCK. And I'll come to that in the next slide because it was key to our initial discovery of Nef inhibitors. Activation of host cell kinase signaling pathways promotes infectivity, transcription, and viral replication. Another important class of effector molecules for Nef include endocytic trafficking molecules, which are responsible for downregulation of cell surface or cell membrane proteins. MHC-1, which I mentioned, as well as viral receptors like CD4 and the infectivity restriction factor called serine-5. So this arm of Nef activity promotes immune escape as well as infectivity of new virions. Okay. So Nef is a tricky protein for drug discovery because it's not an enzyme, it lacks an active site. And so we had to come up with a strategy that allowed us to find small molecules that bind to Nef. So to do this, we took advantage of its ability to bind and activate the host cell kinase known as HCK. So we are able to make recombinant Nef in purified form. We can also make the recombinant kinase partner. As shown in the slide, it's made up of a kinase domain indicated by the K, as well as regulatory domains SH3 and SH2. And 3 and 2 bind the kinase domain and suppress its activity. However, when it binds to Nef, two things happen. One is that it displaces these regulatory interactions. And then secondly, because Nef is a dimer in membranes, it dimerizes the kinases, and together those features allow for kinase activation and signaling. So by modeling this in the test tube, we were able to build a high throughput screening assay where we can show that activation of HCK requires the presence of Nef, and then we ran the screen under those conditions. Okay. So this initial screen, which was published more than 10 years ago now, the reference is here if you're interested. We screened about 200,000 compounds and found about 300 or so hits. And then importantly, we weeded out compounds that just blocked the kinase. And from that, we got about 60 compounds that required Nef to show inhibition of kinase activity. And from that, we identified a series of small molecules and showed by SPR that one in particular, B9, which has this structure, the structure in the box, bound directly to Nef. So from that, we made first generation analogs, and we were actually able to show that not only did this compound interfere with viral replication, it also was able to rescue cell surface MHC-1 in HIV-infected cells and trigger host immunity. So this was a very important proof of concept that Nef inhibitors, chemical Nef inhibitors, have the potential to allow the immune system to see virally infected cells and kill them. Okay, so the next step in our development pipeline was to take this initial hit compound, B9, and create a suite of analogs and try to improve potency and pharmacological activity. And so this was done in collaboration with Fox Chase Therapeutics Discovery in Doylestown, Pennsylvania. So they were our medicinal chemistry partners for this and for the PROTACS, which I'll come to in a few minutes. So here you can see sort of a color-coded version of the original hit compound, B9. It has a central hydroxypyrazole, and then there are 3 arms coming out, red, green, blue. And the chemists were able to make quite a diverse series of about 200 analogs for further testing. So this is where SPR played a big role, because we needed to find or rank the analogs both for binding as well as for antiretroviral activity. So I'll spend a little bit of time on our approach here. Okay, so what was important is to realize that just because a compound binds to Nef, that doesn't mean it's going to necessarily block its function. So we came up with a dual set of screens, one that looks for inhibition of infectivity of the virus, And this is relatively, if you're not familiar, a common assay in the HIV field. It's a luciferase-based readout where if you infect cells with HIV, the initial part of the life cycle leads to activation of a integrated LTR that drives luciferase. And then in the same series of plates, we can also look for cytotoxicity. And then the other part of it is to look for direct binding to the target using recombinant purified Nef proteins and SPR. So the way this was done is we immobilized Nef on the biosensor chip, and then we'd flow each inhibitor over a range of concentrations. And from that, we can get binding kinetics and affinity. And I'll show some representative examples here in a minute. The instrument we use for all of this work is a Reichert 4-SPR instrument, and that's performed very well over the years in this project. Okay. All right, so here's some examples of different behaviors of some of these compounds binding to recombinant Nef by SPR. So here's first example, the compound 7098. You can see this is sort of a typical sensorgram, time base in seconds, and then SPR response in arbitrary units. So we inject the compound, follow its association to steady state, and then stop injecting and follow its dissociation. And then from that, we can fit the curves and derive kinetic constants for on and off rates, and from that calculate a KD value. The other way we look at it is resonance time, and this is the inverse of the off rate. So in this particular example, we get a calculated KD that's picomolar, and the resonance time in relative terms is relatively short, and the extent of binding is relatively high. So here's another example. Again, in this case, the extent of binding is lower, short residence time, but also if you fit this, a fairly high KD value. And here's another analog. This one binds more slowly, but its off rate is quite slow. So there's strong binding, high extent of binding, and a slow dissociation, so long residence time. And then finally, some compounds show no binding whatsoever. Okay, so we screened over 200 analogs, and then we came up with a simple formula to integrate activity against the virus in the infectivity assay that I mentioned, along with the SPR data. So this is one way that we analyze the data, just taking the log of the resonance time, multiplying it by the extent of binding, N for normalized RU at 30 micromolar compound, and then times percent infectivity inhibition. And then we took all those values and then did a z-score analysis, and here's the result for all the compounds that were analyzed. And you can see that out of 217, 164 were active in both assays, and 4 compounds stood out in that they were essentially 3 standard deviations above the mean in terms of this activity score. So these compounds are shown in the next slide. Whoops. I hope you're seeing the slides. Okay. Okay, so this shows the actual SPR traces for the 4 most active compounds from that initial screen. And you can see that all of them bind with very high affinity in vitro and have relatively slow release from the target. And then subsequently, these were assessed in HIV replication assays in peripheral blood mononuclear cells. So these are donor cells infected with HIV-1, and they're actually all able to suppress Nef-dependent enhancement of viral replication with IC50 values in the, in most cases, low nanomolar range. So the IC50s are shown on the slide together with CC50 as a measure of cytotoxicity. And so the two values are compatible with further development. So we also wanted to look at the critical question of whether these compounds could influence MHC-1 display, because that, obviously there are many drugs that suppress HIV replication, but none that can help restore MHC-1 and reactivate the immune response. So we did this in transfected cells, and we found that these compounds showed some activity. So in this plot, if you just look at the plot on the right, this shows the percent of rescue for some of the most active analogs. They're all statistically better in most cases than the original hit compound B9, but still relatively modest rescue. So the question then becomes, well, how could we increase efficacy and actually shut down all Nef functions? So most recently, we've been working with these binders to create proteolysis targeting chimeras or PROTACs for Nef. So this slide just illustrates the concept. So the idea here is that we take the Nef binder and then we connect it through a flexible linker to a second drug. So these are IMIDs or thalidomide-like molecules. And the bifunctional drug molecule allows for recruitment of Nef to the host cell E3 ligase, which contains cereblon. And so what happens as a consequence is that the small molecule induces proximity of Nef to the E3, and then ubiquitin is incorporated into Nef, followed by a polyubiquitin chain, and this allows for degradation through the proteasome. So there's theoretically two benefits here. One is, of course, that you're destroying this viral virulence factor, but at the same time, you're also chopping it up and creating peptides that could then be displayed by MHC-1 to stimulate the immune response. So conceptually, this is appealing because a Nef degrader may actually reverse all known functions of Nef. So this just illustrates this in cartoon form. This is some of the things that I talked about earlier, where an HIV-infected cell initially displays MHC-1, CD4, CCR5, the viral receptors, the restriction factor serine-5, as well as kinase molecules. However, once Nef is expressed, these receptor proteins get downregulated, the kinase is activated, and actually the immunomodulatory protein PD-1 can be induced. So all of these changes help the virus, and I'm not going to read through all these, but you can see that getting rid of Nef potentially may have great benefit. And that's sort of shown here, whereby when the PROTAC comes in, it triggers Nef degradation, potentially restoring all cell surface receptors, shutting down kinase activity, and downregulating PD-1. And of course, generating Nef peptides for immune response. Okay, so how did we make the PROTACs? And so here again, we work with Fox Chase. Our chemist, Colin, took existing Nef binders, and then the first step was to figure out where to attach the linker so that it didn't interfere with binding to Nef, to the target protein. And since we don't yet have a crystal structure or a picture of the binding site, that was done empirically. So he designed a series of 8 or 10 analogs with linkers attached at various points along the molecule. And then using SPR, we asked whether particular positions interfered with binding activity. And from that, we were able to elucidate binding sites at the para position on the two phenyl rings, sort of on the right-hand side of the red structure. So these were then combined with a series of linkers and then bound to a series of ubiquitin E3 ligands. The ones that turned out to be most active involve thalidomide-like molecules, although we also made some for the VHL E3 as well. Okay, so we next initially used a nanobret-based assay to see if these PROTACs could actually induce attachment of ubiquitin to Nef. So this cartoon illustrates the assay. This is an off-the-shelf assay generated by Promega. It's pretty common in this field. So to do this, we fused Nef to nanoluciferase, and then we co-express it with a halo tag version of ubiquitin. And then we added the PROTAC, and then in the presence of the E3 ligase, this would attach the halo tag ubiquitin to Nef. And then when you add the luciferase substrate, you get a signal of 460, so bioluminescence. And then because of proximity, that can stimulate the halo tag, which emits at 618. And so you can look at this in a ratiometric way. And then these are just two examples. Controls in this would be halo without ubiquitin. You can see those are very low. And then when you have ubiquitin tag with halo, there's background ubiquitylation going on, which is interesting, and we're following that up. But when you add a PROTAC, you can increase that in a significant way. And so these are just two examples of actives. So we had about 100 analogs that were synthesized. Most were for cereblon E3s, about a quarter of them VHL, and we found that all the actives are actually linked to cereblon. And then this is a plot, again, just a z-score analysis of all the data, and that allowed us to pick the top 10% or so that were most active for further analysis. So the next step then was to use a system that allowed us to induce Nef degradation in a T cell background. So just to go back for a second. So this assay is done in transfected 293 T cells, whereas this is a CD4 positive T cell line. So this represents a more relevant cell type to look at Nef and Nef inhibitors. So these cells were engineered to express a Nef GFP fusion protein under the control of a doxycycline inducible promoter. And so what this plot shows is as you induce Nef GFP expression, you can follow that by flow cytometry. And then in addition, we stain the cells for cell surface CD4. And so what is hopefully clear from this plot is that as we increase the amount of Nef, the amount of CD4 goes down. And so if we run this experiment in the presence of PROTACs, we are actually able to show both by flow, which is the next slide, and also by Western blot, that the PROTACs are very effective at degrading Nef in this system. So just to go through it, a representative blot is shown at the top. The controls are on the left, and these are just whole cell extracts, Western blotted for Nef expression or for actin as a loading control. So if we don't add dox, there's no Nef. if we add just the co-solvent, which is DMSO, plus DOX, we get a strong band for Nef. And then if we do that in the presence of these PROTACs, so each numbered lane is one individual structure, and these are all active in the nanobread assay that I explained previously, you can see that we get near complete degradation of Nef. And importantly, if we then look at receptor rescue, we can show nice rescue of both CD4 as well as MHC-1 for most of the actives. So here's just a flow cytometry example just to show you the data. It's a 2D plot with CD4 on the y-axis and Nef GFP, so Nef expression on the x-axis. So no dox and no drug, high for CD4, so that's the upper left. And next to it, if we add dox and induce Nef, we see GFP signal, so Nef expression, and then as that with higher Nef expression, you see loss of CD4. If you do the experiment in the presence of an active analog, it looks like the control, so we completely lose downregulation of CD4 in the active cells. And then finally, as a control, this is an inactive analog, and you can see it has no impact. So this bar graph then just summarizes the results for a number of compounds. Here we're looking at CD4, that's the red bars, and we're also looking at MHC-1, return of both receptors to the cell surface. And you can see for for about 6 of these analogs, we get better than 50% rescue at just 3 micromolar. And then in the PROTAC field, it's common to report what's called a DD50. So this is the concentration of drug that's required for 50% degradation of the target. And we did this by Western blot analysis. And for this particular analog, 14369, you can see it's very effective in rescue of both receptors. That its DD50 is submicromolar, and it doesn't show significant toxicity at that concentration. Okay, so getting back to SPR, we were also interested to look at the kinetics of active versus inactive compounds. And one thing we noticed that was interesting is that active compounds seem to have a greater residence time on Nef compared to inactive compounds. So this is just a particularly striking example. So if you look closely at these two structures, the only difference between them is in the linker. So the one on the left, the linker is in black, and right where it joins the Nef binder, it's there's an ether there. And in the inactive analog, it's just a carbon linkage. So that's the only difference between these two compounds, yet their SPR behaviors is quite different. So 14369 binds relatively slowly, but then it remains bound at least over this time scale. On the other hand, the inactive compound shows very rapid association, but then rapid dissociation. And so this kind of makes sense if you think about what's going on with a PROTAC-based drug, right? Because you have to have binding to the target protein, binding to the E3, and they have to kind of get together in the right way for sufficient time to introduce that first ubiquitin moiety. Okay. Also, something I neglected to mention, I don't have a slide of it, but all of the PROTACs were screened by SPR, and not only against Nef, we actually have a whole collection of Nef proteins that are representative of all the major clades of HIV-1 that are responsible for the global pandemic. And the actives, the most active leads, bind to each of those Nef proteins. So that's very important because it's suggestive of broad spectrum activity. The other side of the coin is, well, what about binding to cereblon? So the protein in the E3 complex that is responsible for recruitment of Nef. And we were also able to demonstrate that by SPR as well. Another way to do that experiment too is to look for ternary complex formation. And we can do this in vitro using, excuse me, analytical SEC. So first, please focus on the upper left panel where it says no PROTAC. So this is a plot of protein absorbance as a function of retention volume or time, if you will. We're working here with recombinant proteins. So we have the recombinant Nef alone, same one used in the SPR studies. We have cereblon alone. So it's actually a portion of cereblon that's responsible for binding to the thalidomide moiety, and then the two together. So if you look at the profiles, the molecular weights of these are very close, and so they're not resolved when you run them together. So Nef alone in orange, cereblon alone in red, and then the combo in green. Now, if we pre-incubate with different concentrations of active PROTACs, the structures are shown over here on the right, we get a new peak, and the height of that peak is proportional to the stoichiometry of the PROTAC that's added to the mix. So the experiment is to take the two proteins, add the PROTAC, pre-incubate, then run it through the SEC column, and then look for new peaks. And we see that with all three of these active compounds. And then if we, not shown, but if we take the new peak and concentrate it and then run it on a gel, you can see that both proteins are there. So at least in vitro, through both SPR as well as this approach, we're able to demonstrate assembly of a ternary complex by the PROTACs. And what we're working on now is attempting to use X-ray crystallography to determine the structure of that, because that will help us in terms of structure-guided improvement of these for in vivo analysis. Okay, so do PROTACs inhibit the virus? And the answer is they do very well. There are two assays shown on this slide. So on the left, this is the infectivity assay I mentioned earlier, where we use luciferase reporter cells. And this is a short-term assay where we just infect in the presence or absence of, in this case, an active versus an inactive analog over a concentration range shown at the bottom. And you can see that relative to control, that the active compound blocks infectivity with an IC50 value similar to the DD50. And then over on the right, this is replication again, looking in donor PBMCs infected with HIV-1. The controls are, here's DMSO. So this is wild type HIV, no drug. Next to it is a mutant form of HIV which does not express Nef. So under these conditions, you can see it's about 3 or so fold reduction in replication efficiency. And then if we take wild type and then we add in the PROTACs, we get a block down to, in most cases, what we see with delta Nef mutant. So that's suggestive that the PROTAC is indeed interfering with Nef and leading to its destruction. Okay, so that's kind of all I have. Whoops, I have to do it this way. So where are we with this? So the next real step here is to take the actives and look at their pharmacokinetics, metabolic stability, both in vitro and in mice. And then from that, we can run in vivo assays for efficacy using what are called humanized mice. So these are mice that are bred to be immunodeficient, and then they're reconstituted with human immune cells that allow for both HIV replication, but also have key components of the antiviral immune response. And from that, we hope to be able to establish that PROTACs can indeed suppress viremia in vivo and also prevent viral rebound. We're also interested in mechanistic questions. The Nef variant that we use for most of our studies has multiple lysine sites, and it would be good to know whether one or multiple sites are involved, because this could impact the ability of the virus to escape PROTAC action. And I mentioned the crystallography is ongoing, and then also common in the field is to use unbiased proteomic approaches to look for off targets in terms of degradation in our T cell model. So let me finish by, I want to acknowledge the people that did all the work, the royal we, it's not me. So this is the group that has done all the work that I presented today. So Lori has developed and run all the assays for the initial screening and then ranking of both the Nef binding compounds and then subsequently the PROTACs. Jeff is our X-ray crystallographer, and he's developed the SEC assay that I mentioned and is also working on the structure. Hai-Bin is our expert with SPR, so he's developed all the methods and also screened literally thousands of analogs at this point. Sherry is developing humanized mouse models, and together with our technician Lee. So they are a team that work together for animal studies. Okay, so with that, I'll finish up, and I'm happy to take some questions.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many questions as we have time for. So let's go ahead and get started. Our first question asks, how was the Nef protein immobilized on the SPR chip?
Yeah, that's a good question. I didn't mention that. So routinely we use carboxymethyldextran chips that we get from Reichert. So the attachment is covalent, and it's through the lysines that are on the surface of the chip.
Okay, excellent. Thank you. Our next question here asks, does attachment point or orientation of the Nef protein affect interaction with the small molecules?
We don't think so. I think the Nef protein itself, this particular variant, has 11 lysines, and they're distributed sort of randomly across the structure. So you can imagine that multiple orientations would be presented to the compound, and obviously some of them are compatible with binding activity. There would be ways to address that issue. For example, if you used NTA chips, which can recruit the protein through a his tag, then you would have a known or a fixed orientation of the molecule. But we haven't really tried that, just because the standard immobilization method has been very successful.
Excellent, thank you. All right, our next question asks, how do the Nef binding compounds inhibit Nef function?
Yeah, this is an interesting point, and some additional background, which I didn't get into today, is, well, I did mention it briefly in describing our screening assay, but in cells, Nef, it lives in the membrane, and in the membrane it forms homodimers. And we've shown this using a number of techniques, fluorescence complementation assays, even with biophysical methods using model membranes. And what we found is that both in solution assays as well as in at model membranes, when the binders attach to Nef, they actually interfere with homodimer formation. So that could explain the mechanism for the binders in terms of how they work.
Wonderful, thank you. It looks like we have one more question here in the queue. So if you have questions, this is the time to submit them. Go ahead and put them into the Ask a Question box beneath the presentation. We'll go ahead and ask this one here. This question asks, did you face solubility issues with the binders? And if yes, how did you resolve? Any comments or suggestions about using specific buffers in the SPR assays for DMSO compound, e.g. acetate buffer versus chloride buffer?
Yeah, this is a very good point. And the compounds, some of them don't have great solubility, so we always have to include DMSO. And the concern, of course, is, well, what does the presence of DMSO do to the target protein? So we've looked at this carefully. We use thermal shift assays, if you're familiar. So just essentially asking whether the presence of DMSO has a big impact on the thermal melting point of recombinant Nef in vitro. And we're sort of lucky in that way. The molecule can take up to about 5% DMSO. Now, we don't use that. I think we use 1%, and that works with assays that the highest concentration, I may have breezed over this, but we start at 30 micromolar, and then we go all the way down to submicromolar. So yes, it's an issue, but that's how we get around it. And the buffer, I see the rest of your question there. We just use HEPI's buffer, physiological pH, and then add in DMSO into everything.
Wonderful, thank you. All right, we have one more question come in. This question asks, are all Nef inhibitor candidates tested intravenously, or have there been any nebulized vapor experiments?
So we haven't done a whole lot with the mouse model yet. We have one result with an active binder, not a PROTAC, where we were able to show that in a humanized mouse model infected with HIV, that if we chronically expose the mice to one of the compounds, we can actually prevent or suppress viral replication. The way it was given, again, it goes to the other question about solubility. That was difficult to formulate some of these compounds. So instead, we put them into osmotic pumps, which we then grafted into the mice, and that gave 4 weeks of exposure to the drug, no toxicity, and viral suppression. Since then, we've kind of shifted our focus now to the PROTACs, so we're still at an earlier stage. But once we're able to identify compounds with reasonable aqueous solubility, we're hopeful we can identify one with good oral bioavailability and then administer the compounds that way.
Excellent, thank you. And that is all of the questions that we have today. So we would like to thank LabRoots and our sponsor, Reichert Amtech, for underwriting today's educational webcast. And we'd like to thank you, Dr. Smithgall, for your informative presentation. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for or those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Great. Thank you. Bye-bye.